Welcome to the deep dive. Today we're really getting into the weeds, peeling back the layers on the molecular core of human health and disease, the genetic control of cell function and inheritance.
Yeah, and our sources here, drawn from Porth's Pathophysiology, they really define the biological rules of the road, the mechanisms that dictate not just our traits, but also our susceptibility to all sorts of complex illnesses.
It's essential stuff.
Absolutely.
Right.
Because genetics isn't just some abstract theory anymore. I mean, think about the Human Genome Project wrapped up back in 2003. It fundamentally confirmed that pretty much every common disease we grapple with, cancer, diabetes, cardiovascular issues, they all have a significant genetic component.
Right. So our mission today is to kind of cut through that complexity.
Exactly. Map out the whole system for you. We'll go from the molecular blueprints right up to the tools we actually use now to manipulate them.
Okay, so to give everyone a clear structure, we're looking at four main themes. First, the code itself, DNA and RNA.
Then how that code gets turned into actual proteins.
Right, the expression. Then how all those instructions get organized into chromosomes and passed along through inheritance.
And finally, we'll touch on some of the really cutting-edge gene technology that's shaping personalized medicine.
Think of it as the cell's operating manual.
Precisely. And understanding how that manual is written, how it's packaged, how it's executed, that's the absolute starting point for grasping altered health states.
Okay, let's unpack this, starting right at the foundation. DNA structure and function. We know DNA is the long-term archive, right? Stable.
Stability is definitely its defining feature. It's that classic double-stranded helix structure, perfectly built for storing information and replicating it.
And the structure relies on that base pairing.
That's the key. It's incredibly precise. Adenine always binds with thymine A with T, and guanine always binds with cytosine G with C. They form, like the steps of a spiral staircase, held together by hydrogen bonds.
Okay. And compared to DNA, RNA is more like the working copy.
Yeah, exactly. It's single-stranded, uses a slightly different sugar, ribose instead of deoxyribose, and swaps out thymine for uracilio.
So it's less stable, but that's kind of the point.
It is. That instability works because RNA's job is rapid protein synthesis. So basically, DNA directs, RNA executes.
Got it. But DNA is incredibly long, right? You mentioned 2 meters earlier.
Yeah, roughly 2 meters of genetic material packed into 46 chromosomes in every single nucleus. It's a huge packaging challenge.
How does it even manage that? That seems like a major control point in itself.
It absolutely is. DNA doesn't just float around. It combines with proteins and RNA into this tightly coiled structure called chromatin. It wraps really tightly around proteins called histones.
Like thread around a spool.
Kinda, yeah. And if that DNA is tightly folded, the genes on it basically can't be accessed. They're switched off.
So to turn a gene on?
The cell needs something called chromatin remodeling. Basically, chemical tags like acetylation or methylation get added to those histones, loosening the coil and making the gene accessible for transcription. It's physical folding used as a dimmer switch, essentially.
Fascinating. And the end result of all these instructions? The proteins.
Right. The complete set of proteins encoded by the genome is called the proteome. And the study of those proteins, proteomics, is huge because proteins do most of the actual work in the cell. They're also the targets for most drugs.
But the copying process isn't always perfect, is it? Errors happen.
They do. Mutations can happen spontaneously, or they can be caused by environmental things, chemicals, radiation.
So we just accumulate errors.
Well, thankfully, no. The cell has really sophisticated DNA repair mechanisms. Special enzymes, endonucleases, recognize distortions in the DNA helix, snip out the damaged section.
And then fill it back in.
Exactly. DNA polymerase fills the gap, using the undamaged strand as a template. It's usually very effective.
But if those repair genes themselves are faulty...
Then you've got a problem. Mutations can start to accumulate much faster, and that is a well-established route towards cancer development.
It's also where individuality comes from, though, right? That slight variation.
Absolutely. We share something like 99.9% of our DNA with every other human. It's that tiny 0.1% variation, these differences called polymorphisms, that accounts for all our individual traits and differences in susceptibility to disease.
Okay, so that leads perfectly into how we get from the stored code to actual action, from genes to proteins.
Right. We need to talk about the RNA players involved in executing the plan. There are 3 main types, all synthesized in the nucleus.
Okay, first up is messenger RNA, mRNA. That carries the instructions out.
Correct. It's the template, carrying the code from the DNA in the nucleus out to the cytoplasm where proteins are made.
Then transfer RNA, tRNA.
tRNA acts like the delivery truck. It reads the instructions on the mRNA and brings the correct amino acid to the assembly site.
And the assembly site itself is made of?
Ribosomal RNA, rRNA. It combines with proteins to form the ribosome, which is the actual machinery, the factory, for protein synthesis.
So the first step is getting that mRNA made. That's transcription.
Yes, transcription. It happens in the nucleus. The DNA double helix unwinds just at the needed gene, and an enzyme called RNA polymerase binds to a specific starting point, the promoter region.
That contains the Tata box sequence.
Often, yes. And it makes a complementary mRNA copy of one of the DNA strands. But, and this is really interesting, that first mRNA copy isn't quite ready to go.
And he's processing.
Right. It contains extra bits, non-coding regions called introns that need to be cut out. The parts that actually code for the protein, the exons, are then spliced together.
Okay, introns out, exons kept.
But here's where the cell gets incredibly clever. This splicing process isn't always the same. By varying which exons are included or excluded, a single gene can actually produce a whole variety of different mRNA molecules.
Ah, so one gene can make multiple proteins.
Exactly. Take the tropomyosin gene, for example. It can be spliced in maybe 10 different ways, producing different forms of the protein needed in different types of muscle or other cells. It generates huge functional diversity from a limited number of genes.
Wow. Okay, so once you have that mature spliced mRNA, it leaves the nucleus.
Correct. It travels to the cytoplasm for the next step, translation.
And this is where the code is read, the codon.
Precisely. The code is read in triplets, 3 RNA bases at a time. That 3-base unit is a codon, and it specifies which amino acid should be added next to the growing protein chain.
There are 64 possible codons.
64 combinations, yes. But they only code for 20 different amino acids, so the code is redundant or degenerate.
Meaning multiple codons can code for the same amino acid, like AAA and AG both mean lysine.
Exactly like that. It provides a bit of a buffer against mutation errors.
Makes sense. So the mRNA meets the ribosome.
The RNA machinery, yes. And the ribosome moves along the mRNA, reading each codon. Then the tRNA molecules, each carrying its specific amino acid, match up to the codon.
Delivering their cargo.
Right. And the amino acids link together, one by one, forming a long polypeptide chain.
But that chain isn't a functional protein yet, is it? It needs to fold correctly.
Critically important step. Post-translational processing. That polypeptide chain has to fold into a very specific, complex, three-dimensional shape to become functional.
And that doesn't always happen automatically.
Often it needs help. There are specialized proteins called molecular chaperones that assist in the folding process, preventing mistakes and aggregation.
And if those chaperones fail, or the folding goes wrong?
That's where we see direct links to disease. Misfolded proteins often become denatured, insoluble. They clump together, forming aggregates called inclusion bodies.
And that's the mechanism in diseases like Parkinson's or Alzheimer's.
Precisely. The accumulation of these misfolded protein aggregates is a core pathological process in many neurodegenerative diseases, including Huntington's as well. It's a direct failure of that protein assembly and quality control system.
That connection is so stark. Okay, before we move to chromosomes, how does the cell decide when to even start this whole process? How is gene expression regulated?
Good question. Gene expression isn't always on. It can be turned up, that's induction, or turned down, that's repression.
And what controls that?
The main players are proteins called transcription factors. These are crucial. They bind to specific regions on the DNA, near the genes they regulate.
And they can either help or hinder the RNA polymerase.
Exactly. Some transcription factors recruit the polymerase, boosting transcription. Others block access, repressing it. This is fundamental to cell specialization.
So that explains why a neuron and a liver cell have the same DNA but do totally different things.
Absolutely. They express different sets of transcription factors which turn on or off the specific genes needed for their unique functions. Same blueprint, different sections being read.
Okay, that makes sense. So we've got the code, the players, the process. Let's zoom out now to how this massive instruction manual is physically bundled and passed on. Chromosomes and cell division.
Right. So humans normally have 46 chromosomes arranged in 23 pairs. You get one chromosome in each pair from your mother, one from your father.
And 22 of those pairs are the autosomes.
Correct. The 22 pairs of autosomes. They're homologous, meaning they carry genes for the same traits, though the specific versions, the alleles, might differ.
And the 23rd pair determines sex.
Yes. The sex chromosomes. XX for females, XY for males.
You mentioned something interesting about the X chromosomes in females earlier, the lion principle.
Ah, yes, the lion hypothesis, or X inactivation. In females, early in embryonic development, One of the two X chromosomes in each cell gets randomly inactivated. It condenses down and becomes largely silent.
So different cells might use the maternal X while others use the paternal X.
Exactly. This makes females genetic mosaics for genes on the X chromosome. It's a fascinating dosage compensation mechanism.
Okay. Now, when cells need to divide, there are two main ways, mitosis and meiosis.
Right. Mitosis is how your regular body cells, your somatic cells, divide. It's for growth, repair, replacement.
The result is two identical daughter cells.
Identical, yes. Each daughter cell gets a complete identical set of 46 chromosomes, 23 pairs.
But meiosis is different. That's for reproduction.
Yes, meiosis is special. It only happens in the germ cells, the cells that produce eggs and sperm. The goal here is different, to reduce the chromosome number by half.
So the resulting egg or sperm has only 23 chromosomes, one from each pair.
Precisely. Each gamete gets a single set of 23 chromosomes. So when sperm fertilizes egg, the full complement of 46 is restored in the offspring.
And meiosis is where genetic variation really gets introduced.
Hugely important aspect. During the first meiotic division, meiosis the first, something called crossing over happens. The homologous chromosomes pair up very closely.
And they swap segments.
They literally exchange segments of their chromatids. This shuffles the genetic deck, creating new combinations of alleles on each chromosome that didn't exist in the parent. It's a major source of genetic diversity.
And the outcome differs for males and females.
It does. In males, meiosis produces 4 viable sperm cells. In females, it's unequal. One large, mature ovum gets most of the cytoplasm, and 3 tiny polar bodies are formed, which usually degenerate.
Okay. Now, if there are problems with chromosome number or structure, how do we actually see that? You mentioned karyotypes.
Yes, cytogenetics is the study of chromosome structure. A karyotype is basically a photograph of a cell's chromosomes, usually taken during metaphase when they're most condensed.
And they arrange them in order.
Right. They're sorted by size, shape, and banding pattern. We classify them based on where the centromere, that pinched-in part, is located. Metacentric, middle; submetacentric, off-center; acrocentric, near the end. We also label the short arm P and the long arm Q.
And clinically?
Clinically, it's invaluable. You could just look at the karyotype and count. For example, seeing 3 copies of chromosome 21 instead of 2 immediately diagnoses trisomy 21 or Down syndrome. You can also spot deletions, translocations, other structural issues.
That leads us perfectly into understanding how these traits, normal or altered, are passed down. Patterns of inheritance. We should probably define some key terms first.
Good idea. Let's start with the basics. Genotype versus phenotype.
Okay, genotype is the actual genetic information, the sequence.
Exactly. It's the genetic makeup stored in the DNA base, pairs at a particular locus, or location on a chromosome.
And phenotype is what we actually see.
Right. The phenotype is the observable trait, physical, biochemical, physiological, that results from the genotype. But importantly, sometimes different genotypes can produce the same phenotype.
Like with dominant and recessive traits?
Precisely. We also need alleles. Alleles are the different versions of a gene that can exist at the same locus.
So for eye color, maybe a brown allele and a blue allele.
Correct. If an individual has 2 identical alleles at a locus, say AA or AC, they are homozygous for that trait.
And if the alleles are different,
Then they are heterozygous.
and a heterozygote for a recessive trait is called a phip.
A carrier. So someone who is AA for a recessive condition doesn't show the phenotype because A is dominant, but they carry the recessive A allele and can pass it on.
This sounds like classic Mendelian genetics. Dominant, recessive, Punnett squares.
It is. For single gene traits, Mendel's laws are remarkably predictive. We use dominant alleles, capital letters, and recessive alleles, lowercase. A Punnett square is just a simple grid to visualize the possible combinations of alleles from 2 parents.
Like showing how 2 heterozygous brown-eyed parents could have a blue-eyed child.
Exactly. It shows the probabilities in that case, a 25% chance for the recessive phenotype.
But most human traits and diseases are more complicated than that, right?
Oh, definitely. Most are not simple single gene traits. Many are polygenic, meaning they're influenced by multiple genes at different loci, each having a small additive effect.
And when you add environment to that,
Then it becomes multifactorial inheritance. That's the combination of multiple genes plus environmental factors. Think heart disease, diabetes, many cancers. They fit this complex model. There are other patterns too, like epistasis, multiple alleles. It gets complex fast.
now, one of the really counterintuitive things is genetic imprinting. It challenges the idea that moms and dads' genes always contribute equally.
It absolutely does. Genetic imprinting is a fascinating exception, where certain genes are stamped or marked imprinted based on whether they came from a mother or the father. This marking leads to the gene being expressed only from one parent's chromosome, while the copy from the other parent is silenced.
And the clinical example is striking, chromosome 15 deletion.
Yes, it's the classic example. If a child inherits a specific deletion on chromosome 15 from their mother, they develop Angelman syndrome, characterized by severe developmental delay, seizures, and a happy demeanor.
But if they inherit the exact same deletion from their father,
They develop a completely different disorder, Prader-Willi syndrome, characterized by hypotonia, obesity, and mild intellectual disability. Same deletion, different parent of origin, drastically different clinical outcomes. It highlights an incredible layer of genetic regulation.
amazing. Okay, that brings us to our last major area, the incredible advances in gene technology.
Yeah, this is where things have really exploded, largely thanks to the Human Genome Project. We mentioned genetic mapping earlier, assigning genes to specific loci.
And the big surprise was fewer genes than expected.
Right, only about 30,000 or so protein coding genes. And again, that 99.9% shared DNA. The real action for variation in disease susceptibility lies in that 0.1% difference, particularly in single nucleotide polymorphisms, or SNPs. We usually just call them SNPs.
SNPs are just single base pair differences between people.
That's it. A single letter change in the DNA code at a specific position. There are millions of them.
And the HapMap project mapped these.
Yes, the HapMap project aimed to map common patterns of these SNPs. SNPs that are physically close together on a chromosome tend to be inherited together as a block, called a haplotype.
So you don't need to check every single SNP.
Exactly. By identifying certain informative SNPs within a haplotype tagging SNPs, researchers can efficiently scan the genome to find associations between particular genetic variations and susceptibility to complex diseases, or even how someone might respond to a specific drug. This is foundational for personalized medicine.
And beyond mapping, we can now manipulate DNA, recombinant DNA technology.
Right. Recombinant DNA refers to combining DNA sequences that wouldn't normally occur together in nature. The basic technique involves using restriction enzymes like molecular scissors to cut specific DNA sequences.
You cut out a gene you want.
And then you insert that fragment into a cloning vector, often bacterial plasmid, a small circle of DNA. You put that modified plasmid back into bacteria.
And the bacteria become little factories?
Little factories, exactly. As the bacteria replicate, they also replicate the plasmid and express the inserted human gene. This is how we produce crucial therapeutic proteins like human insulin for diabetes, growth hormone, factor VIII for hemophilia, erythropoietin for anemia. It's been revolutionary for treatment.
There's also DNA fingerprinting?
Yes, based on the same restriction enzyme technology. Because everyone's DNA sequence is slightly different, those SNPs, cutting DNA with specific enzymes produces fragments of different lengths. When you separate these fragments by size using electrophoresis, you get a unique banding pattern for each individual.
Like a genetic barcode.
Pretty much. It's incredibly powerful for forensic identification, paternity testing, much more precise than older methods.
Okay, looking towards future therapies now. Gene therapy and gene silencing.
Two really exciting frontiers. Gene therapy is the idea of introducing a healthy copy of a gene into a person's cells to compensate for a defective one. Or maybe introducing a gene that inhibits harmful process.
How do you get the gene in?
Often using modified viruses like adenoviruses as delivery vehicles. They're engineered to carry the therapeutic gene instead of causing disease. It's been tried for conditions like cystic fibrosis, some cancers, but getting the gene into the right cells safely and effectively and ensuring it integrates correctly remains a huge technical and ethical challenge.
And gene silencing is different, RNA interference.
Right. RNA interference, or RNAi, is a newer approach. Instead of adding a gene, you're trying to turn off a faulty one. You use small, specially designed RNA molecules called small interfering RNA, or siRNA.
How do they work?
They bind to the messenger RNA produced by the faulty gene and trigger its destruction before it can be translated into a harmful protein. You're silencing the gene at the mRNA level.
So you stop the unwanted protein from ever being made.
Exactly. It holds enormous potential for treating diseases caused by overactive or unwanted genes, including viral infections like HIV or hepatitis C, potentially even some cancers. The big hurdle, similar to gene therapy, is efficient delivery of these siRNA molecules into the target cells.
It really ties everything together.
Yeah.
From the DNA code to the potential for targeted intervention.
Absolutely. If you pull it all back to the patient context, DNA stores the information. Chromosomes organize and transmit it. Transcription and translation build the functional parts, the proteins. Understanding how gene expression is regulated, how proteins fold correctly with chaperones, and what happens when these processes go wrong. That's the key to linking these fundamental genetic mechanisms directly to a patient's symptoms and their overall altered health state.
So we've covered a huge amount today, from the DNA double helix as the stable code, RNA as the versatile executor, chromosomes as the organized carriers, the intricate rules of inheritance, including things like imprinting, and finally, the incredible technology that lets us map, understand, and even begin to manipulate our own genetic blueprint.
It really underscores how genetics underpins so much of physiology and pathophysiology.
Right.
We are genuinely moving into an era where this knowledge becomes increasingly predictive and personalized.
Which brings us to a final thought for you, our listeners. Given the accelerating power of SMP mapping to predict risk and drug response, and the potential of tools like RNA interference to custom silence faulty genes, how close are we really to a world where every single drug dose, every therapeutic decision is precisely tailored to your unique 0.1% genetic difference? Something to ponder as you integrate all this foundational knowledge.